Okay, so I want you to just step into a scene with me for a second.
I'm ready.
It's the middle of summer, let's say, late July. You're in a garden, maybe it's a little overgrown,
Mm-hmm.
and you reach down and you pull a tomato right off the vine. It's still warm from the sun, and you pop it into your mouth.
Oh, yeah, that is the perfect bite.
That's the moment,
Yeah.
right? That little pop of the skin, and then this sensory overload. You get that explosion of sweetness, but also that bright, sharp, acidic tang. And it's all wrapped up in this deep green scent that just floods your senses.
It's pure magic, concentrated summer, like you said.
It is summer. Now, fast forward 6 months.
Uh-oh.
It is the dead of winter, January. You're in a big, bright grocery store.
I know this story.
And you pick up one of those tomatoes. It's perfectly round, perfectly red. It looks like a 3D rendering of a tomato.
The infamous red orb.
Yeah.
Hard as a rock.
You take it home, you slice it open, and it smells of absolutely nothing.
Water.
And you eat it, and it tastes like, I don't know, cardboard. Damn cardboard.
We have all been victims of the cardboard tomato.
So here's the mystery that we are going to dive into today. How can these two things, this sublime, magical, vine-ripened fruit, and this bland winter ghost, how can they even be the same species?
Right.
What went wrong? How did this flavor tragedy happen? And, how are we using science to fix it? That's our mission.
And you're right to call it a tragedy, because this wasn't an accident, it was a choice.
A choice.
A trade-off.
Yeah.
It's the story of how, for about 70 years, the global food system basically engineered the flavor right out of the tomato.
Okay, let's unpack that. Because when you look at the history of, say, agricultural breeding, especially after World War II, flavor was not high on the priority list.
Not even in the top 10. It wasn't even on the list.
Really?
No, you have to think about the context. The goal was efficiency. It was about feeding a huge, growing population. So breeders were laser-focused on traits you could measure and sell.
Okay, so let's use an analogy. If you're building a character in a video game, what were the stats they were trying to max out for the tomato?
Oh, that's perfect. They were maxing out defense, inventory size, and durability. That's it.
So defense meaning disease resistance.
Exactly. Disease resistance. Then yield, just sheer quantity. They wanted uniform size and shape, so they would pack neatly in a box.
Right, the inventory part.
And the big one, the most important one, was shelf life. It had to be a tomato that wouldn't get squishy or rot on a truck ride from California to New York.
So in leveling up all those stats,
Yeah.
they didn't realize they were spending points from another category.
Precisely. They leveled up defense and durability, and oops, they accidentally reduced charisma and attack power to 0.
The flavor.
The flavor, it became the genetic casualty because, a farmer gets paid by the pound, not by the amount of deliciousness they deliver.
And there's one really famous example of this, right? The green shoulder problem.
Yes. This is villain number 1 in our story. It's the perfect example of putting looks over substance.
So what was the green shoulder?
So older tomato varieties, before the 60s, they didn't ripen all at once. The part up by the stem, the shoulder, would stay green a little longer than the rest of the fruit.
Which I imagine if you're a shopper, you see a tomato that's red and green, you probably think it's not ripe yet.
Exactly. It looked mottled, inconsistent. So commercially, it was a problem. So breeders went looking for a solution.
And they found a genetic one.
They found a mutation. It's called the uniform ripening locus. They bred this into commercial tomatoes, and what it does is it knocks out a gene called GLK2.
And the result was?
A perfectly uniformly red tomato every time. It looked amazing, a visual masterpiece.
But the cost of that perfect red color was huge.
Devastating. It was a molecular disaster because it turns out those green shoulders weren't a flaw, they were a feature.
A feature? How?
Well, before the fruit was totally ripe, those green parts were packed with chlorophyll. They were basically little solar panels.
So they were still making energy for the fruit.
Yes. Right up until the last minute, they were pumping out photosynthetic products, the building blocks for sugars, and all the complex flavor compounds.
Oh, wow.
So when we bred in that uniform ripening gene, we basically shut down the tomato's last-minute sugar factory.
So in the quest to make it look perfect, we fundamentally made it less sweet and less flavorful.
That's the trade-off in a nutshell. We chose a pretty face over a great personality.
And that was just the first villain.
That was just the beginning. Once you have this blander, tougher tomato, the industrial supply chain adds 2 more layers of flavor destruction.
Okay, villain number 2.
Long-distance shipping. Think about it. You can't ship a soft, fragile, perfectly ripe tomato 1,000 miles. It'll turn to mush.
Right.
So virtually every tomato you buy in a supermarket was picked when it was rock hard and completely green.
I always just assumed they kind of ripened on the truck.
Well, that's the trick. They don't ripen naturally. They get to a warehouse and they get sprayed with ethylene gas.
Ethylene. That's a plant hormone, right?
It is. It's what plants use to signal ripening. But when you spray it on a fruit that's been cut off from the plant, it's just a cosmetic treatment.
What do you mean? What's missing?
When a tomato ripens on the vine, it's constantly drawing sugars and nutrients from the plant. It's a living connection. That's where all the complexity comes from. The ethylene gas just tells the fruit, hey, turn red now.
It doesn't tell it to create more sugar or flavor.
Nope. The sugar factory is closed. The gas it paints the outside red. It's like putting a masterpiece sign on a blank canvas. It looks the part, but the work hasn't been done.
Wow. Okay, so we have the genetic trade-off, the fake ripening. What's villain number 3?
Villain number 3 is the one we are all guilty of. The final nail in the flavor coffin, the cold storage trap.
Refrigerator.
The refrigerator. The science on this is it's brutal. Storing a tomato at fridge temperature around 4 degrees Celsius causes immediate and often irreversible damage.
But we put all our produce in the fridge to keep it fresh.
I know. But for a tomato, it's molecular sabotage. Researchers found that within just 3 to 5 hours of hitting that cold temperature,
3 to 5 hours, that's nothing.
it's nothing. The production of most of those key aroma molecules, the things that actually create the flavor, just plummets.
And does it come back if you take it out and let it warm up?
And that's the truly crazy part. For the most part. No.
No way.
The volatile production does not return to its original levels. You have permanently damaged the fruit's ability to create flavor.
So the fridge doesn't just pause things, it actively kills the flavor.
It actively kills it, and as we'll get into, it chemically locks those flavor genes in the off position. It's a crime against cookery.
Okay, so we've established the tragedy. It's bleak.
It is.
Now let's talk about the quest for a solution. If you want to put the flavor back, we first need to know the recipe. What actually makes up that perfect tomato flavor?
Okay, so flavor is basically a triad. A 3-legged stool, you need sugars for sweetness, you need organic acids for that tart, tanny note.
Right, the sweet and sour.
Exactly. But the third leg, the one that does all the heavy lifting, is volatile organic compounds, VOCs.
The volatiles. The invisible flavor agents.
They are everything. Without them, a tomato is just sweet and sour water. The VOCs are the aroma molecules. Our brain interprets that smell as tomato flavor. No smell, no soul.
And scientists have actually mapped all these out, right?
Oh, yeah. They've identified something like 227 different volatile compounds. They group them into categories like grassy, fruity, floral, woody.
That sounds complicated.
It is, but we
Yeah.
can simplify it. Let's just focus on the 2 main molecular chefs that are running the flavor kitchen.
Okay, let's meet chef number 1.
Chef number 1 is in charge of the fatty acid-derived volatiles.
Oh, what?
These are the green notes, that sharp, fresh smell you get when you, brush against a tomato plant.
Oh, I know that smell. It gets all over your hands.
It's the one. The key compounds are things like hexanal, and they come from the breakdown of fatty acids in the fruit.
Okay, so this is where it gets a little technical. Let's use that chemical factory analogy you mentioned.
Yeah.
How does this process work?
Perfect. So step 1, you need the raw materials. An enzyme called lipase goes and releases the fatty acids.
Okay.
Step 2, another enzyme, a lipoxygenase called Tom-LOX-C, comes in and oxidizes them. It adds oxygen.
Got it.
Step 3, another enzyme acts like a molecular shredder, chopping these oxidized acids into smaller pieces, these little C6 aldehydes. That's the grassy smell.
And you're saying that's a huge part of the overall smell.
It's a huge part of the volume. These compounds can make up over 80% of the total volatiles.
Wow.
But, and this is the key, more isn't necessarily better. Studies show that these grassy notes aren't what people actually love most about the flavor. Too much of it is just sharp and green.
So 80% of the smell isn't 80% of the enjoyment. There has to be a final step.
There is. This is the crucial finishing touch. A final enzyme, ADH2, comes in and converts those sharp aldehydes into alcohol.
That changes the smell.
Completely. It softens it.
Right.
It turns that raw green smell into a much more pleasant, ripe fruit aroma. The best tomatoes run the whole assembly line, not just the first few steps.
Fascinating. Okay, so that's the green grassy chef. Who's molecular chef number 2?
Chef number 2 is in charge of the apocarotenoid-derived volatiles.
Okay.
These are the good ones, the fruity, the floral notes, things like biionone and geraniolacetone. This is the perfume of the tomato.
And where did these come from?
This is the beautiful part. They come from the breakdown of carotenoids.
Wait, carotenoids? Aren't those the pigments, the things that make the tomato red in the first place?
Exactly. Lycopene, beta-carotene. The molecules that give the fruit its color are also the raw material for its most delicious fruity smells.
So the color is literally the source of the flavor.
It's an incredibly elegant system, and researchers have found that these apocarotenoids are way more important for whether people actually like the tomato. If you're missing these, it just doesn't taste right.
So we have the recipe book. We know the ingredients. The problem is that the industrial field is a terrible kitchen.
Terrible kitchen. It's chaotic. Too much rain, not enough sun, weird soil. It's completely inconsistent.
Which brings us to the solution. The controlled environment of the greenhouse.
The greenhouse is the superpower. You can control everything. Temperature, humidity, CO2, and most importantly, you can let the fruit ripen fully on the vine because you're not shipping it 3,000 miles.
Right, but the control goes even deeper than that, especially with hydroponics.
Oh, much deeper. In a hydroponic system, you are in complete control of the nutrient solution. You are basically a chemist fine-tuning the flavor.
So give me an example. What kind of levers can you pull?
Okay, let's say you want a tartar, more acidic tomato. You just increase the amount of potassium in the water.
It's that direct.
It's that direct. You are literally turning up the tartness dial. But then you have to be careful with nitrogen. Too much nitrogen, especially early on, gives you what growers call bullish growth.
Bullish growth, what's that?
It's all leaves and stems, no good fruit. The plant puts all its energy into growing big and green instead of making delicious tomatoes. So you have to keep nitrogen low at the beginning.
You're forcing it to focus on the fruit factory, not the leaf factory.
Exactly. And then there's calcium. You need high calcium for firm, juicy fruit. But
There's always a but.
there's always a but. The high potassium you use for tartness can actually block the plant from absorbing the calcium.
Oh, no.
And that leads to blossom end rot, which is this horrible black spot on the bottom of the fruit. It's a constant balancing act.
A chemical tightrope. You can't just adjust one thing without thinking about everything else.
Right. It's why these high-tech greenhouses are full of sensors and AI constantly monitoring and adjusting.
But the most exciting dial you can turn is light.
Light is the master switch, the intensity, the spectrum. It controls everything about flavor production. This is how we can scientifically replicate a perfect summer day any day of the year.
Okay, so let's compare. What does low light do versus high intense light?
Okay, so think of a struggling winter crop. Low light, low temperature. That environment pushes the plant to make more of those grassy, fatty aldehydes.
The sharp green notes.
Exactly. The plant is stressed, you get a thin-tasting tomato, the chemical factory is underperforming.
And the opposite.
High temperature and intense light that supercharges the breakdown of those red carotenoid pigments.
Which gives us...
more of the good stuff. The fruity, floral, apocarotenoid volatiles, you get a much more complex, delicious flavor.
So the perfect summer day literally tells the plant's chemistry to make the flavors we love most.
And now, with LEDs, we can create that perfect day on demand. We can use specific combinations of, say, white and red light to optimize the sugar-acid ratio. We can dial it in.
That's incredible. It's a complete reversal of the industrial trend.
We can finally optimize for flavor and consistency, not just for shipping.
So we've covered the environment. Let's go back into the DNA. To really fix this for the future, we have to find those genetic keys we threw away. How did scientists even start looking?
Well, the first problem was that their map was incomplete. For decades, the entire world of tomato genetics was based on one single reference genome.
One tomato.
One variety, yeah. Heinz 1706, the one they use for ketchup.
Why is that a problem?
It's like trying to understand all of humanity by only studying one person. Modern tomatoes have incredibly narrow genetic diversity compared to their wild cousins. By only looking at the Heinz genome, we were blind to thousands of genes that were lost during domestication.
So we're using a heavily edited guidebook.
Exactly. The solution was to build a pan-genome.
A pan-genome.
Yeah, instead of sequencing one tomato, they sequenced 725 different kinds. Cultivated ones, heirlooms, and critically wild relatives. They built a massive, comprehensive map of every possible tomato gene.
And what did that new map show?
It was staggering. They found 4,873 genes that weren't in the original Heinz reference at all.
Almost 5,000 lost genes.
5,000 lost genes just reappeared on the map. And it immediately showed them which genes had been lost during domestication. A lot of them related to disease defense and, you guessed it, flavor.
And this led them back to that one enzyme.
It led them right back to tomloxy, the one that kicks off the green, grassy smells.
Right.
Using this huge new map, they could zoom in on the control switch for that gene, the promoter region.
The part of the DNA that tells the gene when to turn on and off.
Precisely. And in that switch, they found a rare version, a rare allele.
An allele is just a variation of a gene.
Right. And this rare version seems to have been accidentally thrown out a long time ago when farmers were just selecting for big, high-yielding fruit.
We tossed out the flavor switch.
We did. But here's the really cool part. They found that modern tomatoes that have one copy of the common version and one copy of the rare version.
The heterozygotes?
Yes. Those tomatoes had much higher expression of the flavor enzyme than tomatoes that had two copies of either one.
Wait, so it's not about having the good rare one, it's about having one of each.
It seems to be about balance. Having one of each appears to create the perfect level of enzyme production at just the right time during ripening. The answer wasn't to replace the old gene, but to bring back its long-lost partner.
That's the DNA sequence. But there's another layer of control, right? The dimmer switch. Epigenetics.
Yes, so epigenetics. These are chemical tags that sit on top of the DNA. They don't change the code itself, but they tell the cell which genes to read and which to ignore.
So if DNA is the sheet music, epigenetics is the conductor.
That's the perfect analogy. And for a tomato to get ripe and flavorful, the conductor has to tell the ripening genes to play loud. It does that by removing some of these chemical tags in a process called demethylation.
The tags silence the gene, so you have to remove them to turn it on.
Exactly. And now we can finally connect this back to villain number 3.
The refrigerator.
The refrigerator. We now know that chilling stress, putting a tomato in the cold, causes rapid changes to these epigenetic tags.
So, when I put a tomato in the fridge, the cold is telling the conductor to put all those silencing tags back onto the flavor genes.
You are literally telling the flavor orchestra to go silent.
Wow.
That is the molecular reason why cold kills flavor. You are triggering a rapid and often permanent change in the epigenetic dimmer switch.
So the foundation is set. We know the history, the chemistry, the genetics. Let's talk about the future. What tools are we using now to rewrite the tomato's story?
The number one tool is CRISPR, genetic editing. It gives us a level of precision that old-school breeding could only dream of.
So with traditional breeding, if you wanted a disease-resistant gene from a wild tomato, you might accidentally bring along a gene for bad flavor that was right next to it.
That's called linkage drag, and it was a huge problem. But CRISPR is surgical. It's like using a find and replace function on a specific word in a document, instead of cutting and pasting the whole messy paragraph.
So what can we do with that kind of precision?
Well, one of the first targets was sweetness. Researchers used CRISPR to knock out one tiny little regulatory element in a gene called SLBZIP1.
And what did that do to the fruit?
A huge increase in the amount of natural sugars. They made the tomato fundamentally sweeter just by editing this tiny little break that was holding back sugar production.
That's amazing. And what about that original sin, the trade-off between shelf life and flavor? Can CRISPR fix that?
It already has. This was a huge breakthrough. Scientists used it to knock out 2 specific genes that are responsible for the fruit getting soft and mushy.
And the result?
A much firmer tomato with a way longer shelf life, but crucially, with absolutely no negative effect on the flavor compounds.
So we can finally have both, durability and deliciousness.
The compromise is over.
That's incredible for the lab, but outside the lab, we have a massive problem. Climate change. We need a tomato that can survive the heat.
Absolutely. The projections are terrifying. We could lose a huge percentage of our yield for every single degree of warming.
So where is the plant's biggest weakness when it comes to heat?
It's reproduction. The most sensitive part is the growth of the pollen tube.
Okay.
If it's too hot, the pollen tube fails to grow properly, and the plant can't set fruit. It effectively becomes sterile.
High heat just shuts down the whole factory.
It does, but again, we look to the wild relatives on our Pan-Genome map. Varieties from hot climates, like the Tamaulipas tomato, they have a natural molecular armor against heat.
And we can figure out what that armor is and give it to our commercial tomatoes.
That's the goal. They're even looking at developing sprays with small molecules that could, tell the pollen to put on its heat armor right before a heat wave.
Molecular defense on demand.
Exactly. And they're making direct genetic fixes, too. In one study, they inserted a tiny bit of DNA into the promoter of a sugar gene.
And what did that do?
It increased the yield by almost 50% under normal conditions. But under severe heat stress, that edited plant recovered more than half the yield that the normal plant lost.
So we're building the ultimate tomato. Tastes like summer, lasts for weeks, and can survive a heat wave.
That's the mission. It's a complete reversal. We went from accidentally losing flavor to intentionally designing it for a new climate.
It's wild to think that this whole flavor disaster wasn't malicious. It just happened because we didn't understand the science. We didn't know what a VOC was or why one little gene mattered so much.
Exactly. And the real irony is that the most artificial environment, the high-tech, climate-controlled greenhouse, is now the only place we can perfectly replicate the ideal natural conditions for the best possible flavor.
Okay, so we've armed our listeners with a ton of knowledge. What is the one practical, immediate takeaway they should have from this deep dive?
It's simple, and it comes back to that chilling injury. We now know, without a doubt, that refrigeration actively destroys the flavor of a ripe tomato. It scrambles the chemistry and rewrites the epigenetics.
So the final word is never put a ripe tomato in the fridge.
Never. If it's ripe, keep it on the counter. We are in the middle of this incredible scientific war to bring back great taste. It would be a real shame to have all that effort undone by an appliance in your own kitchen. And that leaves a final thought for you to chew on. If we know for a fact that the cold sabotages something as complex as a tomato, what other simple habits in our kitchens might be silently wrecking the effort to bring us delicious food? Now you know the science. Keep them out of the cold.